Hello, my name is Jeff. I'm here to talk about mining of PubMed abstracts. I did present on a paper recently about Zaremba et al., and I felt that that article wasn't so great, so I decided to look for more articles related to mining of PubMed abstracts. So, why is this paper significant by Chen et al.? Well, the paper by Zaremba et al. Only had 12 citations, but this article had 281 citations, so I wondered what the difference was, and that made me curious, which guided me to reading this specific article. Of the 281 citations of Chen's paper, yes, maybe some mentioned that it was just a tool, but there are several studies that actually used the tool itself. You can see some of the names of the publications here that included, or actually using ChiliBot. So I thought that was very interesting and something to keep in mind of why the Zaremba et al. Paper was not great, but this one is much better. So this article by Chen et al. Developed a cool tool called ChiliBot. It stands for Chip Literature Robot. The purpose of the ChiliBot is for a rapid identification of relationships between any entities of interest. It's very nice for a large number of queries. It can speed through it very quickly. And it's free to use. There are 3 different types of search options: 2 terms, pairwise, and 2 list. The 2 terms process is simple. You just look for a relationship between 2 words, 1 word and 1, 2, it finds the relationship. Pairwise, as you can see from the picture here, right to the right, at the top is the top is the pairwise, and then the bottom one is the 2 list option. And you can see the diagram of how it looks differently, or looks for the relationships between each of the words. On the right-hand side is a screenshot, and that's just from my work a few days ago. I took that screenshot. So how does ChiliBot work itself? It's on the left. You can see it looks like a pipeline. First, you give ChiliBot a query. It could be anything. Genes or free-form words. For example, apoptosis could be a term that you search for. So you enter those queries, and then it collects the abstracts from PubMed. Now understand that there is a 30 abstract limit. Well, why can you only use 30 abstracts? It seems a bit low. Later I will show you that really the number of abstracts is not really too terribly important. Once it collects all the abstracts, you have to look up synonyms. So, for example, we have a query for apoptosis. The system will also look for cell death. Now, the synonyms come from an internal database collected from 6 different databases. On the top right, you see those databases that they were collected from, the 6 databases. And then parsing is the next step. You have to parse out the sentences from the abstract to individual sentences. Once you have the individual sentences parsed out, then there will be parts of speech tagging. You can see what I mean here on the right, the bottom right, the CAAS, etc. Once this information is parsed out, an NLP rule is used to classify the relationships. Stimulation, inhibitory, neutral, parallel, or co-occurring. Once the relationships are identified, then you just have to show that information to the user using an AISEE, which is a third-party company. Now, to discuss the methods. How you go from abstracts to sentences. First, a sentence has to be identified, having both query words inside that sentence. And then it's used for tagging. Suppose you have a sentence that does not have both terms in the sentence, that sentence is discarded. So then you use tagging for the parts of speech. Realize that parts of speech are important because they help us identify verbs. For example, if you have just two nouns, like cat and dog, we don't know what's going on. It's a cat and a dog. Suppose you say the cat ate the dog. Oh, great. Now we know that something's going on here. So, in biology literature, it's not a cool topic like that, like something's eating something, but you have, like, for example, activate, induce, and block are verbs. You also have to search for passive voice and negations, and then use weighted terms to find and identify the different parts of speech. Once the identifications happen and the NLP rule can be extracted for the relationships, and then this is a large picture of the relationships. And the boxes that are blue, as you can see here, like cocaine and plasticity, the two bottom blue boxes on the screen, those were the two query words. Then it creates a graph with different terms that could be involved in that interaction between cocaine and plasticity. Now, what does colorblind mean? So the green means, what does color means? The green means stimulation, and the red means inhibition. And there's a key here on the left where you can get more information. The lines show the relationships between the terms. I did look at this and notice that it doesn't seem to be colorblind friendly. That's just an interesting critique I had for this specific diagram. Now, once you evaluate the performance, how do you do that? So there are 770 known relationships through the interaction database, and it's good recall, 90.5% recall rate, which is pretty good. I did notice that there were not a lot of comparisons to other competing minors. There wasn't really a comparison, which I didn't like. I would like to see more of comparison between this tool and others. Now, there were 68 failures that happened with this process, and the main reason is that the general protein names, like pKa, does not provide enough information. There are different kinds of pKa. For example, it's not specific enough, seems to be the issue, one issue. So that may be a problem with the abstract itself or the system. We're not quite sure. Now you can see partial results. On the right, figure 3b, you can see the item number of abstracts increases and the number of relationships doesn't really increase from that, which means it seems that the number of abstracts doesn't seem to impact the number of relationships. On the left-hand side, you can see the number of sentences that increase, and then the numbers of relationships decrease. But as the number of abstracts used increases, the number of relationships also start to be skewed to the right. This is just more showing that the amount of abstracts used doesn't really impact the amount of relationships found. As long as the number of abstracts is not above 30, which is the limit for this system. This spreadsheet here, this graph, shows why the failures typically happened. Typically because the names were too general or incomplete synonym list. There's an incomplete synonym list. Those are a few reasons that I noted. A really cool part of the ChiliBot is the hypothesis generation. It looks for undiscovered common knowledge. So what is that exactly is this hypothesis? It just says that A causes C. But really, you have to look further into that. So A affects B, and then B affects C. The goal is to find the relationship between A and B, the relationship between B and C. If you can find those two relationships, then you can say A might impact C for these specific reasons. We gave it a query of 22 genes involved with LTP. It's a neurological process that you don't really need to worry about. It's not important for our knowledge. Just know that it's already well known. So with all of the literature, they have found all the relationships. Oh, well, that's what should happen. All the literature should find all the relationships. But now we're traveling back in time, so to speak, using a retrospective study that limits LTP to just before 1995 or 1990. You still can find relationships, even limiting to your information to retrospective studies. This table is a bit confusing, I will say. The third column you see here is what you give to the ChiliBot query. Then you would be able to find the relationship to LTP through the, so knowing A to B relationship and the B to C relationship. The third column is the the second column is the B relationship. The third column is A. Really it goes column 1 is C, column 2 is B, and column 3 is A. So the last few columns show that the limited literature to before 1995 still allows you to find relationships, which is very cool. So what if this is all just random noise? We have to notice important characteristics of the relationships and to show a very interesting shape. It's called a scale-free topology. On the top here, the left picture is a random network, and you can see it's just absolutely random, nothing special about that. On the right here at the top is a scale-free version, and you can see several hubs that have been established. That means some nodes have various connections connecting to one node, and that's a characteristic of many models from nature. For example, social media. Now, using random selection gene to try and find the relationships, we created this picture that's on the bottom. And you can see that there are many scale-free. I mean, it looks scale-free, which is a good thing. It means it's similar to what is actually occurring in nature. Now, the number of relationships. As the number of relationships per node increase, the probability decreases, which makes sense because you're not able to find a node with many, many relationships. So that makes sense. In mind. There are my references for your review. Do you have any questions? Thank you for listening.